Hi, we're NYMU-Taipei 2011 iGEM team from Taiwan National Yang-Ming University, with tailoring to avatar being our project this year. Okay. So the story begins with a magnetostatic bacterium named Magnetospirillum magneticum strand AMB1. Okay. These bacterium contains organelles called magnetosomes. These magnetosomes are comprised of magnetite mineral crystals that enables the bacterium to sense magnetic field. Turns out throughout iGEM history, there are totally 16 iGEM teams who have proposed or tried to apply the magnetostatic bacterium in their project. While most of the team has faced difficulties on bacterial cultivations in that it takes 1 week for the bacteria to reach a plateau. Wow, okay. And a lack of platform to manipulate this bacteria. Except for 2 teams, there are 2060 who has operated the bacteria as an AC generator and as NYMU-Taipei team, being the only one who has actually provided solid evidence and a complete toolbox to iGEM, including a new vector, a plasmid for manipulating and transforming genes into AMB1. It is equipped with our RA and RAP gene from PMGT, which is a plasmid found in another strand of magnetostatic bacterium, with an appropriate promoter and RBS clone from AMB1. And these genes are constructed on a POC19 backbone to complete our design of a new plasmid PYMB. Meanwhile, we have collaborated internationally with some of the universities in China to testify the promoters and RBS, and finally get our result to confirm that the expression of the plasmid PYMB does work on AMB1, but not E. coli. Okay, but what does this have to do with the movie Avatar? If you have ever seen it, do you know that the movie was based on the concept of a remotely control of an avatar body? Remember that to become an avatar, one must lie inside a machine which transfer his mind into an avatar body. Since this mind transferring part, this mind modulation part being so fascinating, our team was then wondering if there are any possibilities that we can actually realize this avatar fantasy. After several literature research, we found then promising techniques, optogenetics, by delivering laser beam through an optic fiber cable into the skull of animals. And meanwhile, viruses were engineered to infect nerve cells with special types of channels. And once the laser beam shines, the channel snaps open, iron rushes in, and fires the nerve cell. That is a neuron modulation. This has been named Method of the Year 2010 by Nature Methods. But think cleverly. Are we going to carry this laser beam cable on our head all the time? No, definitely no. This conflicts with the idea of a remotely modulation of neurons. So what should we do? That's why we have applied the optomagnetic bacterium in our experiment, with several modifications have been conducted that it is able to twinkle. And this replaces the laser beam that is a light source in our experiment when sensing magnetic field. Now, let's take a closer look at our general design. First of all, to reduce immune responses, we have enabled our bacteria to reach symbiosis, that is to live with the glial cell. We can then inject the glial cell into the animal brain. Okay, so once we applied a magnetic field to it, our bacteria then started to glow and triggered the downstream reaction of the nerve cell. Yeah, okay. So we have achieved a remote modulation of neurons by applying optomagnetics and neurosymbiosis. Next on, we're going to introduce to you our optomagnetics, the modeling part, and neurosymbiosis, the immune responses we have concerned. And so the Synthoprime, a software tool we've designed this year, and our human practice part. Next, let us welcome our modeling part.
Okay, thank you. Okay, after the introduction of our project, let's see our optogenetic design. We want to applying the magnetic force, and the magnetobacteria will be enlightened, and it will induce the downstream neurons. The selection of the magnetic force is due to the convenience to manipulate. And how about the enlightening part? We want to design one on-end of neuron modulator which can be modulated by the magnetic force immediately. So how to do this? Let's take a deeper look in our bacteria. Here is a bacteria of AMB1, and there is a magnetosome in the bacteria, and which is composed of the magnetites. And here is a modeling image of the magnetosomes. The red particles represent the magnetosomes, and they are aligned on the green cytoskeletons. There are also some special proteins called MF13 on the membrane of the on the membrane. And what they have is they have two helices on the transmembrane domain and one loop tight bonding to the magnetosome. A paper published in 2006 that the authors successfully anchored one luciferase protein on the MF13 C terminus. And based on this exciting research, what we want to do now is that we came up with the wild idea that is to anchor two proteins on both of the sides of MF13. When the magnetic force is applied, they will pull the magnetosome down, and the two proteins anchored on the MF13 will have protein interaction. And thus, the light will emit, and it will modulate the downstream neurons. So what we want to do is use the Breit phenomenon. The Breit, bioluminescence resonance energy transfer, is based on the energy derived from the R-luciferase reaction. When one fluorescent protein gets closer to the luciferase, the energy will excite the fluorescent protein, and it will emit the light. However, due to the original tight interaction between two helices of MF13, it will only cause the on state, but we want to have the on and off both states. So what we do is we make a little adjustment. The first, we chose the YFP as our luminescence protein because the light induced by the luciferase and the YFP exactly fits the wavelengths to excite the neurons. And the other adjustments that we insert one chain peptides that to design to fit with higher affinity with helix 1 than helix 2, that to block the tight interaction between two helices. So the result will be only when the magnetic force is applied, two helices will get closer together, and the YFP will emit the light. However, if the magnetic force is no longer applied, the two helices will have farther distance due to the chain inhibitor. So it will be the off state. And in this sense, we can say that we create an on and off both states modulator of the downstream neurons. So since chain peptide plays an important role in our project, let's take a closer look at how did we design it and our modeling part. There are several sophisticated steps for designing the chain design. So here we will mainly focus on two important parts. One of this is the selection of the template pairs, and the other is the packing side chain energy calculation. The first, we have to know the structure of the MF13. So we use 9 online prediction program to predict the structure of MF13. So here is the structure. Then it's to select the template pairs. To select the template, we have to consider the critical GF3G motif, which has the helical surface, and it can maximize the van der Waals for the interactions between the helices, and it could also reduce the distance between two helices. So consider the GF3G motif and some straits on the MF13. We select one template pair from cluster 2s in the bank reference from paper published in 2006. So then it's the next step to calculation that the energy being minimized. So we base on this function with six energy. We use the online program Yasara Energy Minimization Server for calculation the above 3, and the other is Delta G Prediction Server to calculate the below 3. So here it is a result about our cross modeling. Then we compromise those results for choosing the final choice. And we also choose for the other seven amino acids to fit the chain design. So you might be curious whether it will get in reality. So we use the fridge magnet to pull the magnetotactic bacteria, then we can see it will be put up. So it we have more confidence that our device might work just to carefully modulate the frequency. So we might use the device that we have 3 now for the electromagnet. And if we want to have the higher gradient of the magnet, we can use the advanced one, reference from the paper published in 2004. Okay, let's see our constructs. We have several constructs, but first we have to have the materials that is calling PCR from the magnetobacteria AMB1. That is our construct. We have several positive and negative constructs to infer the misfolding of the proteins or to show that whether the CHAMP design is truly work. And the best one is our ideal construct, which have the YN and YC by using the biaxy braid to improve helix 1 and CHAMP's affinity. So next, let's have our results. We first finished all the construct, then we successfully proved that YP did work and emit the light in the YP MF13 CHAMP or luciferase constructs. So let's welcome another part, our neuron symbiosis.
Hi everyone. For delivering our magnetostatic bacteria into clinical use and inducing optogenetic response, we have also considered immune responses in a trigger. So in order to design our neural symbiosis one step in vivo, first we inject our AMB1 into the pure strain mammalian mixed glial cells. And then we selected the most stable interaction between the glial cells and the AMB1, and we then inject it into the brain. In order to introduce our AMB1 into the brain, there must be many problems we may encounter. The first one, what will be the responses following the injection, and what if our AMB1 grows without control, and how does AMB1 stay within the brain? So we have to establish a symbiotic system with brain. There are 2 strategies we can use, extracellularity and the intracellularity. The reason we didn't choose the extracellularity is because it's too difficult and uncertain for us to prevent the AMB1 from spreading out. And we have also considered the other reason why we chose the intracellularity. The first, we want to avoid exposing bacterial antigens. And it's more precise for us to trigger the downstream nerve impulse. Also, we have reviewed many genetic toolboxes passed in the iGEM. We found that most infectious bacteria like Cox's disease in brain are using the intracellularity. So we chose to establish intracellularity. Let's take a closer look at the 2 main groups of cells situated in brain, neurons and glial cells. We didn't choose neurons is because it may automatically trigger the neuron activity. And it is also difficult to inject into the brain, and neurons themselves are unable to regenerate. How about our glial cells? It is easy to inject and they are able to regenerate. So we can then focus on the relationship between the AMB1 and the glial cells. And then we figure out the ambitious solutions for our neural symbiosis. We utilize 4 different proteins. The first one is invacin, which is a gene that can let our bacteria to get into the glial cells. The second protein we use is LLO, a gene that facilitating escaping from the phagocyte zone. And the third protein we use is ZMP1, a gene for avoid ubiquitination that leads to inflammasome activation. And the fourth protein we use is MinC, a cell division inhibitor. We have constructed these genes from the genomic DNAs of Yersinia, Listeria, and the E. coli K12. These are the 2 constructs we designed for the neural symbiosis. Based on the tetracycline-induced system on AMB1, we use this system to regulate MinC expression. We can see from this figure that the expression of theta protein enabled our regulation. On the other constructs on invacin and LLO, we refer to 2010 Hwasa team and we designed it to be compatible on AMB1. Here's our advantages of one step in vivo. First, take our MB1 into the mixed scalar cells, and it can reduce the pathogenicity. And we hope that we could select the most stable interaction between them, and we can make sure the histocompatibility. So next, welcome our Synthprime.
In our project, we have designed a lot of constructs, and it requires a lot of primers to do PCR. In this year, we are glad we have designed a new software, which is Synthprime. It has expressed 1,400 total genome. So rather than passing the sequence directly, you can choose the organism as well as the gene, and it will bring the sequence automatically to you. And it has also includes the basic parameters of GC content and melting temperature, as well as a ranking of primer products. And together, the convenience of it will definitely prompt the development of synthetic biology. And as we have done an outstanding project, we also want to probe its social impacts of our projects as well as its related SE issues. So in human practice, we have held a grand convention in our school, and we want to introduce a new approach, which can be divided into opening survey, discussion, and closing survey. In opening survey, we found out that 71% of all participants are really high interested in discussing the issue of neuroethics. And in discussion, we want to introduce a brand new strategy, which is called role-playing strategy. This strategy that participants were assigned a role, and they give their opinions based on this role. And after integration of all the viewpoints from different roles, new ideas have formed. And this strategy has worked out really dramatically and has given us a thorough dimension of our main theme, which include neuroethics as well as other topics. We hope it not only works for us, but can serve as a model for all iGEM teams. And now let's welcome our conclusion.
Thank you. So to conclude our project, from this point on, we have already known that we have achieved an unprecedented optomagnetic system as well as a one-step in vivo neurosymbiosis. Another point is to be mentioned that we have set up a chassis that has been the dream chassis for iGEM. And another point that's very important here, that we are in the new application track. So this new application, synthetic neurobiology, was never seen before in iGEM. Let's talk about some other strategies in synthetic neurobiology these days. Be they deep brain stimulation, optogenetics modulation, or even the two-dimensional optogenetics modulation. There are one drawback in common. That is the lack of complexity that we can deliver it to the brain. So we can help to ask one question: is it how synthetic it can really be? So I thought of these problems, and we all together try to solve this problem through some studies, through some inspirations from delicious research. We found out that in this stage there are a system of two-dimensional. Wait, there's a system of sorry about that. Okay, a two-dimensional optogenetic system, as well as a wireless and multi-channel system. And a third one that inspires us the most is the mind-altering bacteria. Combining those three ideas standing on the foundations, and we have come up with this idea that, that we develop an injectable system to replace the implanted electrodes that may cause neurodegeneration in the brain. And in this project, as indicated in the science paper this year, the scar generated from the injectables injection will be repaired by parasites, and this cannot be done in a system that's facilitated by electrodes or even optic fibers. So we have achieved we have to say we achieved a three-dimensional dot matrix stimulatory system that can change the way we think about circuitry in iGEM. What's that? It is about a system that is, we use this system to, there was a film in that. So let's just give it the film. We build a system with several syringes aligned in a matrix. We insert it, we penetrate it, and we inject a little bit of things. We we throw a little bit, and we jog again. We throw a little bit and jog again. So this time, we will be able to construct a three-dimensional dot matrix stimulator system using the the spatially varying magnetic field to probing neural correlates or neural substrates that's having a higher complexity or even the highest complexity of all neural substrates. That give rise to an idea that maybe one day we can imagine that a brain or some mysterious neural substrates or like the neural correlative consciousness and the activities can be recreated. So we have thus named this project the Tailoring Your Avatar. So thanks for listening. Welcome to a new world.
Hello, this is iGEM News. Nowadays, we are facing the problems about the increasing number of population, the greenhouse effect, and environmental pollution. They may be the main problems in this century. Fortunately, a research group is going to solve these problems. They gave an amazing approach that using cyanobacteria to perform endosymbiosis with human cells for conquering Venus. Also, they opened a new leaf for Venus immigration. This group comes from Taipei, Taiwan. They are NYMU-Taipei.
We all know that nowadays there are lots of serious problems in our environment, such as the overloading of population and great amounts of pollutants such as carbon dioxide and sulfur dioxide, and make our Earth worse and worse and become improper for creatures to survive. Therefore, we came up with an idea that maybe we can conquer Venus and become a Venetian. But it occurs to two problems. That first of all, the atmosphere of Venus contains lots of carbon dioxide and sulfur dioxide, and it is apparently not proper for us. And the second problem is that we also need lots of energy and lots of food. So here's our solution. Because it contains lots of carbon dioxide in the atmosphere of Venus, so we decide to use cyanobacteria to start our project. Then we clone two genes, sulfide reductase and sulfide quinone reductase, into our cyanobacteria. Sulfide reductase can transfer sulfur dioxide into hydrogen sulfide, and sulfide quinone reductase can use hydrogen sulfide instead of water as the reducing energy in the photosynthesis. And because our final goal is to become a Venetian, so we also use in-phasing plus lysol-lysing O, which is an executive part from 2010 Warsaw team, and to make our cyanobacteria perform endosymbiosis with our eukaryotic cells. For practical application, besides testing on Venus, we found out that in some industries that the water waste and air pollutants have the same components as the atmosphere of Venus. So we decided to test our cyanobacteria in the factory. So here is the whole picture of our project. First of all, because we use synthesis PCD7942 as our cyanobacteria. Then we use sulfur reductase to transfer sulfur dioxide into hydrogen sulfide. And by the combination of SQR and chloroplast, we can create carbohydrate. And in the future development, that we can use GLUT gene to transfer our carbohydrate out from our cyanobacteria, so that our host cell can use it. We clone sulfide reductase out from disulfide disulfricants and SQR out from synaptose enterococcus PCD722. And based on our in-gene cyanobacteria, we can also put these two genes inside it and perform endosymbiosis in our stem cell.
Okay, hi there. Let's see how it goes on. First of all, we already know that we use the cyanobacteria to perform the photosynthesis for us to use the carbon dioxide to produce energy. But cyanobacteria, they are too weak to use, because they cannot use the sulfur, they cannot change sulfur as the energy. So here we clone two genes. The first one is that sulfur reductase. We take advantage of sulfur dioxide and not only use that as the resource, but to produce the energy for us. So as you can see here, we clone two sulfur reductase. The first one is called ESR, which is from the Desulfovibrio desulficant. It's a kind of sulfur-reducing bacteria. Another one is called CIS1, which is from Pseudomonas aeruginosa. By doing so, we clone those two genes. We can easily transfer the sulfur dioxide into hydrogen sulfide. And then here, we performed a hydrogen sulfide producing assay to test our gene. As you can see, we follow this paper, and we use DPTA as our solution to absorb the hydrogen sulfide. We can easily demonstrate the color change to you. Sorry. So this is the data. As you can see here, this is the standard curve we've done, which confirms the authenticity of our experiment. And right here, the red line, which is our engineering cyanobacteria, the CIS1. Compared to the black control, they can produce much more hydrogen sulfide, right? Which means our gene works and our ideas work too. Okay, let's do the next part.
Yeah, so after the sulfur oxide, okay, got to see. Thanks. So after the sulfur oxide is reduced, energy cell inside the sulfur So, hello. So,
Don't hold it like that. Just put it on the shirt. You will adjust the volume after you ignore it. Please put it there and ignore it.
Okay, so after the sulfur oxide reduced energy store inside the chemical compound of hydrogen sulfide, and sulfide quinone reductase, or SQR, then comes into effect. SQR is able to oxidize sulfur compound into sulfur and release bound energy for further favorable reaction of photosynthesis. And in ordinary photoassimilation, photosynthesis to dissociate water and form oxygen subsequently. Yet in our engineered cyanobacteria, hydrogen sulfide is oxidized and sulfur is produced. In other words, we construct a sulfur version of photosynthesis instead of a traditional water and oxygen version. So to confirm the function of our new biobricks, we conduct two series of experiments. First is the measurement of cyanobacteria growth, and second is the actual sodium sulfide consumption rate in E. coli. In sulfide-containing medium, we found that SQR actually outnumber its wild-type counterparts. And next, we added DCMU, a chemical which can shut down the photosynthesis too. By doing so, we force cyanobacteria to use electron donor other than water, because photosynthesis will block the ordinary electron transport chain. And in such DCMU-containing medium, since SQR is able to use sulfide, hydrogen sulfide, so it prospered as we expected. Yet in wild-type counterpart, just die out. So again, these results suggest that SQR do make a significant difference in sulfide-containing medium. However, although that optimal density, the absorption is a very common method to estimate the growth of the cyanobacteria. However, it just provides us approximation, a rough approximation on the cultural level, but not a quantification on the viability of the cells. Thus, we use a method published last year called the autofluorescence assay method. And from the positive and negative control group, we established, and we found that we must use the PRCP fluorescence states. Comparing the fluorescence signal from both SQR strain and wild-type counterparts, we found that SQR strain, as we expected, has a larger and higher signal and had higher cell counts, while the wild-type ones have a lower fluorescent intensities, just like the negative control group. And we also conduct plate count experiments to gain access to the actual cell counts. We grow the SQR strain above, in the above plate and wild-type one below. And we found that on the same condition, SQR strain actually had higher cell number comparing to the wild-type one. And next is the actual consumption rate of sodium sulfide in E. coli. After 48 hours, SQR already consumed as much as 2.2 times as that of the wild-type counterpart. And this figure shows us another important results. While the SQR used up almost every sodium sulfide added inside the medium, the sodium sulfide added inside the wild-type one simply added up, because wild-type one cannot use sodium sulfide. So the sodium sulfide added inside wild-type one just accumulates. Again, this result reflects the ability and the function of our new biopricks. And although it seems that our project is a good solution for the environmental problem, yet our ambition ain't just end here. So let's welcome our excellent correspondent.
Okay, after solving the problems of the Venus gas, SL2, now let us think about the crazy idea. Is it possible to create Venetians or even to land on Venus? Literature revealed that the engineered E. coli with the invasive gene from Yersinia pestis can invade into the mammalian cells. And the engineered E. coli with the listeria O from Listeria could escape from Ferguson and live intracellularly. An engineered cyanobacteria with the invasive and LLO gene could invade into mammalian cell, the child cell, and the J774 macrophages and live intracellularly. Now let's turn to our project. In our previous experiment, we did a great job on the sulfur metabolism and solved the problems of Venus gas SL2. And now let's how about creating Venetians? Symbiosis is generally referred to a mutualistic relationship where both partners benefits. Thus, once our cyanobacteria could invade into the cells, it is possible for the eukaryotes to survive on Venus. That is to say, to create Venetians. In our experiment, we transformed the invasive and LLO gene into our cyanobacteria and co-culture the cyanobacteria and the mammalian cell together. And 24 hours co-culturing, the cells, J774 macrophage cell lines, and mouse-induced pluripotent stem cells were evaluated. Now, let's turn to our results. Under the fluorescent microscopy, the cyanobacteria could release the autofluorescent red light. That is to say, the red part is a cyanobacteria. And after the 24 hours co-culturing, our unique cyanobacteria could invade into the J774 macrophage cell lines. And in this picture, it also revealed that after 24 hours co-culturing, our cyanobacteria could invade into the induced pluripotent stem cells. Especially the induced pluripotent stem cells are artificially derived from the non-pluripotent cells, and it can differentiate into many kinds of cells or tissues. So it provides more potential for our project to create Venetians. So after realizing the whole project of the conquering Venus, now let's to see the practical use in our project to Earth.
So it sounds like it's a really complicated project, right? So after all, we have to do some, we live in Earth right now, so we have to do some practical experiment to confirm our project. But just as we say, we live in Earth now, not in Venus. And we have no idea about NASA. So if you guys are familiar with NASA, please tell us. But it's very interesting that we find out in Earth several industrial waste has the same component as the Venus, such as the industrial wastewater and waste gas. So we take the advantage of them to test our cyanobacteria. So here we cooperate with this industrial industry. And we simply co-culture our injuring cyanobacteria with the industrial waste to test them. So as you can see here, we still perform the hydrogen sulfide producing assay. This is in the waste gas part. And as you can see here, the red one and green one are our engineering cyanobacteria. Compared to the blank control, they can produce much more hydrogen sulfide, and in both layers, water layer and the gas layer. So which means our gene works and our project works too. In this part, we use the wastewater to test our engineering cyanobacteria. Still, we perform the hydrogen sulfide assay. You can see that the red one and the green one, compared to the blank control, they produce much more hydrogen sulfide. So our project works again. So we integrate those factors, and finally, maybe you just can become the Venetian, and maybe let's go to the Venus. Right.
What an impressive work they did. Amazing of them. Okay, thanks for your time. See you in Boston.
Hello everybody, I'm Venusian. 1 month ago, I was attracted by some aerotypical scattering around Taiwan when I guarded the Earth as usual. After visiting NYMU-Taipei iGEM 10, I realized that this is one of their brand new human practice. So let me introduce what they have done of another human practice to you. They hold meetings within Taiwan iGEM 10, having fun, advocating their project, and advocating synthetic biology field. Now let me tell more detail. By holding meetings within Taiwan iGEM 10, they're realizing other team projects more, created the Taiwan iGEM discussion platform, cooperating notes, common notes in both English and Chinese. And finally, they have collaboration with NTU Taida, and they prove their gene works. And by doing so, they let future iGEMers in Taiwan to discuss and exchange their ideas easier. And they also advocate in their project by making board game and iGEM song and iGEM short film. They also advocating synthetic biology to foreign medical exchange students and our NYMU freshmen. By doing so, they introduce iGEM and synthetic biology to people who never knew them. And after those activities, they add people into NYMU Facebook iGEM Club. And they also plan to shift iGEMers recruiting time earlier and to create an iGEM Club. By doing so, they let people know iGEM, interested in iGEM, and finally they will join us and engage in synthetic biology field.
Okay. Hey, Venetian, why are you here? Please go back to your Venus. It's our time. Live long and prosper. Thank you. So let's sum up. We guys have already created a whole new 11 parts and sent them to the part registry already.
And we have characterized and proved that these fabrics do work out to effective.
And we not only modify an already existing part, which is the LLN invasing, which done by Warsaw University in 2010. And we use them to furthermore to demonstrate them and create a Venetian.
And we held out a meetup with the iGEMers in Taiwan and even helped NTU Taida to solve and identify one of their fabrics.
And finally, we guys really did a good work in the human practice part. As I mentioned that, we guys are the first one iGEM team who try to communicate with the Venetian, such as the aliens and some outer space human, right? So it's a kind of human practice, I think. And after all, we guys really had lots of fun. We had lots of fun by doing this project. So, and right here, we have to thank to those are our acknowledgements. So thanks to them. Okay, finally, we are NYMU-Taipei.
The Father Venusian, thank you.
And thanks for your time.
Hello, everyone. Judges, guests, welcome. And welcome to the 2013 National Yangming University iGEM presentation. This year, we've chose to focus our project on a problem in the world called colony collapse disorder, CCD, which is the rapid deaths and disappearances of bees all over the world. Now, we have an introductory video to introduce you to this problem. But before that, here's a team photo.
I'm a queen bee, and my family is dying, and my colony is collapsing. What is the cause of CCD?
That's a great question. Now, scientists have numerous speculations as to what is causing CCD. However, research and literature points to one primary factor, Nosema cerana. So what is Nosema cerana? Nosema cerana is a microsporidian fungus, which in other words is a unicellular parasite, and it infects the midgut of a bee. And it gets into the midgut through either bee-to-bee contact or water sources. Now above, we have a life cycle of a Nosema cell. On the top right-hand corner, you're able to see a polar filament of the Nosema, which is a needle-like protrusion from the Nosema spore, which is used to inject genetic material into the epithelial cell of a bee midgut, thereby reproducing and infecting the bee.
So, doctor, how are you going to combat Nosema?
That's a great question, and I have a simple answer for you. B. coli. So our B. coli has 5 primary mechanisms. Firstly, it enters the bee. Secondly, it suppresses the development of the Nosema's polar filament. Thirdly, it kills the Nosema. Fourthly, if it seems like the bee's infection by Nosema is past curing, it kills the bee. And fifthly, we've included a light inducible system so that our B. coli only operates inside the midgut of the bee. And all 5 of these mechanisms together combine to form our final product, which I'll talk about in more detail later.
So, can you explain more detail about your mechanisms?
Absolutely. Let me hand over to my colleague, Dr. Ro Chen here, who's going to explain more.
Thank you, Dr. Rohan. The first mechanism we use to cure CCD is to prevent the Nosema infection by prohibiting the germination of Nosema. This is the microscopic image of Nosema spores. Once it gets into the midgut of bees, the polar filament will go out. And if you remember, the polar filament of Nosema spore is a needle-like protrusion from Nosema spores, and it will inject the genetic material into epithelial cells. And it's composed of the mannose polymer. Therefore, we choose to express a kind of protein called FinH, which will bind to the mannose polymer and prohibit polar filament from piercing epithelial cells. To determine whether FinH functions as expected, first, we add FinH-RF fusion protein and pure RF protein into the eppendorfs containing mannose polymer. Secondly, we wash our mixture. As you can see on the result, at the left-hand side, the mannose did bind with the FinH-RF protein and emit red light. Compared with the right-hand side, the RF protein was washed out with the supernatant, so there's no efflorescence here. However, if the infection of Nosema is too serious and the Nosema germination cannot be prevented by the FinH protein, the next mechanism we use to cure CCD is to treat our bee by killing Nosema. We decide to take advantage of the immune reaction of the bees. Once Nosema entering the midgut of bees, the epithelial cells will secrete RS. RS is the abbreviation of reactive oxygen species, which will oxidize OxyR, the transcription factor of RS-induced promoter. This year, we use 4 of this promoter, and the first 2 ones, Himetron and Trixie, are their first time to be introduced to iGEM. Furthermore, we improved the promoter HPCP, which we got from 2010 KIT Kyoto team. HPCP is actually a complicated promoter. And first, we do a point mutation on it to mutate the PST1 site. Secondly, we delete the reverse coding sequence and splice the HPCP to D1 in 3 different ways and made a part collection. And based on the promoter strength and the sensitivity of this promoter, the HPCP to T1 turns out to be the most appropriate one for our project. However, the concentration of OxyR, the transcription factor of HPCP to D1 in native E. coli, is only 10 to the power of negative 15 molar. However, the concentration we need to trigger the HPCP to D1 promoter is approximately 0.005 molar. Therefore, to produce more OxyR, we decide to express it constitutively. And based on our modeling result, if we express it by a constitutive promoter J23102, the concentration of oxyar will rise above 0.005 molar, and it's enough to trigger the HPCP2D1 promoters. And furthermore, we do the promoter testing of HPCP2D1 with and without the constitutive expression of oxyar. Then we see that, as you can see on the result. With the constitutive expression of oxyar, the promoter strength of HPCP2D1 turns out to be 9 times stronger than before. Therefore, we choose the HPCP2D1 promoter as our sensing promoter. And after our B. coli acquired the ability of sensing NOSMA, the next question we confront is, what can be used to kill NOSMA? And based on the literature, we find out that the higher the concentration of antimicrobial peptide, the higher the survival rate of bees infected by NOSMA, which means that the antimicrobial peptide will raise the survival rate of bees. Therefore, we decide to express the antimicrobial peptide defensine in our B. coli. Defensine is functioned by creating a pore in the NOSMA plasma membrane, which will lead to the efflux of NOSMA genome. As you can see on the result, this specific fragment of NOSMA genomes show up after we did gel electrophoresis. This indicates that our defensine was effective in lysing NOSMA spores, and it can kill NOSMA as well. And then we are going to talk about the regulation of curing circuit. I'll hand over to my colleague, Dr. Chen.
Thank you, Dr. Rou Chen. So after introducing our weapon, how are we going to control this weapon through different stages of nosema infection? The answer for this question will be the circuit design. So in a healthy bee, since there's no nosema serrana, so the ROS will not be produced and the sensor will be off. The TET promoter will be on, leading to the production of the CI protein, which will bind to the PLUX-CI hybrid promoter and causing this hybrid promoter to be off. So in this case, our killing protein will not be produced. While in a sick bee, since there's no nosema serrana, there will be ROS produced and the sensor will be on. The TET protein will be produced and binds to the TET promoter, causing the TET promoter to be off, and the CI protein will not be produced. So those CI protein remain in the hybrid promoter will then be degraded. You can see that in the downstream of the sensor, there's a LuxI-LuxR gene, and LuxI protein is an enzyme that will produce HL, which will bind to the LuxR protein and form a complex. This complex will activate the hybrid promoter, causing the promoter to be on. So our killing protein will be produced, and in this case, it will be the defensin. We've also designed a positive feedback in order to enhance the production of the defensin. So after nosema is eliminated from the bee's midgut, there's no nosema, so RS will not be produced, and the sensor will be turned off. So the TET protein, the TET promoter will be on, and CI protein will again be produced and binds to the hybrid promoter, causing the hybrid promoter to be off. So our killing protein will not be produced, and this will go back to the initial status of our circuit design. So the next time when nosema serrana invaded, it will go through the previous process and kill the nosema. So if the nosema infection comes to a point that cannot be cured, then our B. coli will move on to the next step, which will be the sacrificing part. We managed to kill one bee in order to save the whole hive. The way we kill the bee is through using the ethanol. Ethanol can harm the central nervous system and causing the death of the bee. So we've cloned 2 genes which will be the PDC and ADH. It will produce ethanol through the metabolic pathway. So we've come up with the problem that how much amount of ethanol we use that can kill the nosema. So we conducted an experiment called the bees killer run. We've divided the bees into several groups and injected different amounts of ethanol. And the result is 2 lambda of 95% of ethanol can kill, at least this amount can kill the bee. So this will be the circuit of our ethanol parts. And we can see that there's a terminator set right behind the sensor. The terminator set is served as to slow down the production of ethanol so that our ethanol will not be produced when the infection of nosema is not that severe. Once nosema infection comes to a point that it cannot be cured, then it will produce the T7 polymerase, which will activate the T7 promoter circuit and causing the production of the PDC and ADH. We've also designed a positive feedback in order to shorten down the time of the ethanol production and let the concentration can reach the effective level in a short time. So through modeling the concentration changes of ethanol to the time, we can see that through our circuit, the ethanol concentration can reach the effective level that can kill that can kill the bee. So besides concentration, timing is also important in our operation. Since after 120 hours of the nosema infection, the nosema will start to spread to other bees and causing the infection of the other bees and causing the death of the other bees. So all of our operation needs to start before 120 hours. So through modeling the concentration of the both ethanol and defensin to the time, we can see that after 30 hours of the infection, our defensin will be produced and start killing the nosema. And if the infection comes to a point that cannot be cured, at 80 hours after infection, ethanol will be produced and kill the bee in order to save the whole hive. So we can see that all of our operation will start working and before the 120 hours before nosema starts to spread.
So, doctor, have you ever considered about your B. coli accidentally invade into the environment?
Well, this will be our safety part. So we've introduced a light-dependent lysis system. So since we want our B. coli to stay in the bee's midgut, so it will be dark there. So in a dark environment, the lysis system will be off. And when the B. coli comes out of the bee's midgut, then it will have interaction with the light and turn on the lysis system and causing the death of the B. coli, which is our E. coli that will kill the nosema.
So, how can you transport your B. coli into the bees?
Well, let me hand over to Dr. Rohan to introduce our product.
Thank you. So we've created these complex mechanisms and circuits. However, a central problem that all of this boils down to is how are we supposed to get our B. coli into the midgut of the bee safely in the first place? Therefore, we've decided to use a system called encapsulation, building off of the 2009 Imperial College iGEM team. What encapsulation is, in essence, is we have our B. coli, and we're coating the outside with a layer of alginate solution, which serves two primary purposes. Firstly, it protects the B. coli from the immune system, the immune reaction of the bees. And secondly, if our capsules accidentally get into the environment, it prevents and blocks the B. coli expressed proteins from going outside into the environment. We've conducted experiments on this encapsulation method. On the two photos on your left-hand side, you'll be able to see microscopic slices of a bee's midgut. Now, on the bottom two photos, we fed the bee with a capsule which enclosed B. coli expressing GFP. And as expected, during fluorescent microscopy of the bee's midgut, green fluorescence was present. And in the top, we fed the bee with no capsule. And when we did fluorescent microscopy of the bee's midgut, as expected, there was no fluorescence. In fact, we've brought an example of what our final product will look like. Over here.
Is there anything inside these?
No, there isn't. We know. So it's an example of what our final product will look like. We understand the safety. And as you can see, it looks a lot like a sugar bead. The reason is because on the outside of the capsule, we will be able to coat a layer of sugar, and then this layer of sugar would serve the purpose of making it easier for beekeepers to dissolve the capsules into sugar water solution, which can then be fed to the bees. Now, naturally, in different bee colonies, we will have different infection severities of nosema. Therefore, we've decided to compare the infection rate of a certain bee colony to the relative amount of capsule we need to feed to the bee colony to the survival rate of the bee colony. For example, let's say an infection rate of a bee colony is 40%, and we wanted to raise the survival rate to 40%. The relative amount comes down to 5, and this means that we will be able to combine the number 5 and integrate it with the number of bees in the colony to get a definite number of capsules we need to feed to the bee colony.
So, you guys are bravo. And what else have we done in the society?
Great question. Let me answer it right now. In terms of our human practice, we can separate it into 3 main parts: the application, experimental cooperation, and outreach. In terms of application, we went around to different beekeepers in Taiwan, and we've asked them to give them samples of their bees. And we've used a novel nosema diagnosis system that we created ourselves to diagnose these bees. And much to our surprise, as well as the beekeepers, these bees actually were infected by nosema. In terms of experimental collaboration, we collaborated with the NYMU-Taipei team, and we helped characterize some of their parts. And in terms of outreach programs, we presented and promoted synthetic biology in iGEM to the entire freshman and sophomore class of the Yangming National University. We've also went around gathering local beverages from Taiwan and sending them over to Brazil to help and assist the Brazil iGEM team with their project. And perhaps most notably, we've helped establish a high school iGEM team locally called Taipei American School. And thereby, we're bringing synthetic biology into an entirely different realm, that of high school. They will be competing at MIT at the Jamboree next year. And to help such newly established iGEM teams, we've established some tutorial videos in how to do some experimental procedures so that they will more easily be able to learn how to do iGEM experimental procedures. In conclusion, to sum up, here are some of our achievements. We've created 23 new BioBreak parts and submitted them to the registry. We've created a novel way of transporting engineered E. coli into living organisms. We've created a new modeling standard of prescribing engineered E. coli to disease-infested locations. We've also created an inducible positive feedback delay circuit. And finally, we've improved our AHPCP promoter so that it's 9 times stronger than it originally was. And here are some of our accomplishments during the Asia Regional. We'd like to thank all of our sponsors who have supported us in our endeavors. None of this would be possible without them. And here are our references.
Thank you, Doc. Thanks.
No problem. Thank you. Thank you.